All right. Let's jump into it. Our topic today is biological integrity. And we're going to be pretty water focused today, but I think there's a lot of lessons learned for all of the biological needs of our communities. And, water is certainly a very important asset that we have around the state of Minnesota as well. And so there's reason to be focusing certainly on this particular area. So I am going to pass it over to Jill to introduce our agenda and topic a little bit more.
Okay, well, glad to have you all here. Glad to see some people from other cities too. It's good to find out what they're doing. Yes, this is a really important topic, biological integrity. We see declining monarch and rusty-backed bumblebee habitat and species populations, and they are becoming a rallying cry for conservation-minded residents that are establishing pollinator gardens and rain gardens. And reports of declining and collapsing ecosystems is common in the news, so we're more and more aware of that and the relationship with climate change. In our urban areas, the impervious cover on the landscape contributes to flooding and poor water quality. And I see this as firsthand because I serve as a manager on a watershed district board where we have 3 creeks that flow into the Minnesota River. And then along those 2 of the creeks, we've got 13 lakes. And GreenStep cities are contributing data on the efforts. We've got 5 GreenStep cities actually in my watershed district to become more energy efficient and reduce waste. And they're tracking their greenhouse gas emissions well. But how do cities balance development and protections and restoration of the natural areas that the residents value? And what metrics will better inform decision makers to restore healthy lakes, ecosystems, and help mitigate climate change and climate impacts? So those are some of the questions I'm hoping to get some information and guidance from our wonderful speakers today. A little bit about the Izaak Walton League. I don't know, Kristen, if you can throw up the logo that we have. So we are we're local, state, and national. So this is the pledge that we recite every time we get together to start a meeting: to strive for the purity of water, the clarity of air, and the wise stewardship of the land and its resources, to know the beauty and understanding of nature and the value of wildlife, woodlands, and open space, and to the preservation of this heritage. We've got 2 logos up there. We just celebrated our 100th anniversary, and there were like 54 anglers out of Chicago. The black bass they were avid anglers, fishermen. The black bass were dying off. Our rivers were being treated as sewers, and wildernesses were getting fragmented, and they launched our organization. So, but Izaak Walton lived in about 1620 and in England, and he saw the value of healthy trout streams to what the land use was around and adjacent to those streams. So that's why we became the Izaak Walton League. But what's becoming apparent is that what we're learning from the emerging sciences study of ecosystems is that the ecosystems have evolved together. So, and it's particularly important for those. So we've got 15 active chapters around the state, from Detroit Lakes area on down to Winona. So they reflect in their concerns the different diverse biomes of the state, everything from woods and wilderness in the north to wetlands and prairies and oak savannas down to the Mississippi floodplains and bluffs. So this is all really important to those of us, and a lot of our work is done in chapters, supporting our communities and trying to raise these issues. Some of the things that we've accomplished we started in 1922. By 1924, the Upper Mississippi River Wildlife and Fish Refuge was established by the Izaak Walton League, mainly led by Will Dilg. But into 1924, President Coolidge signed off on establishing that. So that was a major accomplishment of the Ikes early on. And there was also they've pushed for preservation of the Boundary Waters Canoe area wilderness, the Tamarack National Wildlife Refuge up in the on the other side of the state from where I am, and even Richard Joydor with the Memorial Hardwood Forest. Those were actually he was a member of our Bush Lake chapter in Bloomington, but was passionate about getting that forest established. We've also supported campaigns to save the wetlands program, and been big supporters of passing Clean Water Act, the Wild and Scenic Rivers Act, Clean Air Act, excuse me, and the Wilderness Act, and others things that are safeguards for conservation and public health. So, the pollution, meaning air and water quality, and support for sustainable communities have long been league priorities in our first 100 years. But what we're realizing is that we've always considered ourselves to be defenders of air, soil, woods, waters, and wildlife. But the emerging science is showing us that the study of these ecosystems, that the nature evolves as a system, and that there are these symbiotic relationships among and between air, soil, woods, waters, and wildlife. So that second logo, which we adopted in July at our annual convention for our launching our next 100 years of work, really kind of you see those symbiotic relationships visualized there. So the question is, how do we go about ensuring the biological health of our natural resources that we value so much? And what is needed to inform natural resource management and climate mitigation in a state that's so rich in these? So hoping to get those answers. We do have some best practices that are part of our program. And maybe Jack can even talk. I don't know if you've done anything with any of those. There are some listed. But a lot of it's about engaging community people. I know we've tried to be there to, like was mentioned before by some of the cities, they need the citizens to be driving this agenda to get this on the radar of city council people. So anyway, but there are, I don't know, Kristen, if you wanted some of those talked about or not, but there are several best practices that would be GreenStep cities can be doing to help ensure the protection of our natural resources or get it on the radar and make sure that there are programs to help enhance that. So
yeah, there are quite a few, and I apologize for so many so many words on a slide. So we will be sharing this out to workshop attendees and with the recording so that you can actually click on all of those and explore some of the best practices in the GreenStep program that relate to biological integrity and habitat conservation in your community. So yeah, thanks, Jill.
Sure. So with that, shall we start with Jacquelyn?
Yeah, so Jacquelyn, I think you're going to share. No, John is going to share the screen, right?
Oh, Sean's gonna, okay.
John's actually going to talk first.
We got a team tag here.
Yeah, I
Gonna
think
really
I'll start
educate
and
us.
then pass it to Jacquelyn. All right. Folks can hear me okay?
Yeah, and if you could just reintroduce yourself quickly for the recording, would be good.
Yep. So my name is John Sandberg. I work with the MPCA's River and Stream Biological Monitoring Program. I'm based out of Brainerd. We have a group of, I think we have 9 biologists here, and we cover the entirety of northern Minnesota. Going down as far south as about St. Cloud, and then we range up to the northern border and cover the Red River Basin and Lake Superior Basin. And then there's another work group out of the St. Paul office that covers the St. Croix Basin and the southern half of Minnesota. So like I mentioned, we work on rivers and streams. We do primarily biological monitoring of fish and aquatic invertebrates, but we also do some water chemistry monitoring to support that effort. Yeah, that's probably a good introduction, and maybe Jacquelyn could introduce herself.
Sure. So I supervise a similar program only for lakes. So we have, I should have counted, I think there's 7 of us that are Fish IBI staff that kind of travel around the state and collect the data. But then we also within DNR utilize our existing fisheries area office structure. So we have about 20 area offices that are also collecting the data. We collaborate to pull all that information together on the biological health. And in addition to doing the direct fish surveys, we actually do a fair amount of habitat survey work as well, which I'll talk about later on.
Okay, so I think the way we've laid this out today is I'm going to spend a little bit of time talking about some broad concepts related to biological monitoring and assessment. And then I'm going to pass it to Jacquelyn, and she will really illustrate these concepts with some specific material about the use of our IBI tools in Minnesota lakes. So obviously, Minnesota is a water-rich state. I think sometimes those of us who have lived here for a long time almost take it for granted, but I know from having talked with folks in other states that we are in a very unique position here, given our abundance of fresh water and high-quality fresh water. Lakes, rivers, wetlands, we've got it all here. We've got lots of it. And, you know, for a while, just the sheer quantity of water in Minnesota presented some challenges in terms of the state adequately meeting our obligations under the Clean Water Act. So the Clean Water Act is obviously, you know, it's been in place for 50 years now. It's the 50th anniversary this year. And this is one of the overarching, you know, fundamental regulatory tools that have allowed us to maintain the chemical, physical, and biological integrity of our water. So for many years, you know, we've been doing biological monitoring at the PCA since the early 90s, and we've been doing water monitoring since, you know, the inception of the Clean Water Act. But it really wasn't until the passage of the Clean Water Land and Legacy Amendment in, I think it was 2008, that we were able to implement a more systematic and comprehensive framework for monitoring our water and trying to develop strategies for restoring waters that need restoring and protecting our high quality waters. So this is really important. I wanted to acknowledge that, you know, the work that we do is directly funded by these amendment sales tax dollars. And it's really provided us with these good tools now that we can adequately move through the state. This is an animation of how our watershed approach played out from 2008 through 2017 as we rotated around each major watershed in the state, doing monitoring work, assessing the water to determine where our impaired waters were and where our high quality waters were, reporting on that, trying to use that information to guide implementation efforts, whether it's restoration or protection. And now as we're starting our second cycle of the watershed approach, we're going back, monitoring a lot of those same places again to see how things are changing. Now, when I talk about monitoring, what exactly is the purpose? So the idea is to determine whether water and aquatic habitat is of sufficient quality that it can be used for all these different things that we need it to be used for. Drinking water, aquatic life, aquatic recreation. There's industrial uses for water, agricultural uses, just general aesthetic enjoyment and recreation. These are big, broad concepts of how water is used that are identified in the Clean Water Act. And then here in Minnesota, we've actually put these into rule. These are what water is, these are the uses for water in Minnesota. And this is how we're going to protect these uses. We have specific water quality standards related to drinking water, and aquatic life and recreation, industrial consumption, and so forth. Now, to talk about each of these would probably be a presentation on its own. And the work that Jacquelyn and I do really focuses on these, what we call class 2 uses for aquatic life. And aquatic life use support is one of the primary objectives of the Clean Water Act. It's an important water quality goal in Minnesota. And this concept of biological integrity relates directly to whether or not water can support aquatic life. So how do we know whether the water is clean enough to support aquatic life? For many years, we had what we consider to be surrogate measures. We measured the amount of suspended solids in the water, the levels of dissolved oxygen, pH levels, chloride concentration. These benchmarks were all established, based on laboratory testing to understand, when levels of a pollutant reach a certain level, how do organisms respond? And those were very effective, in addressing a lot of the water quality problems we had back in the middle part of the 20th century. But as time went on, it became apparent that we needed more direct and comprehensive measures for protecting aquatic life. And that's where these tools, which we'll talk about shortly, which directly evaluate the condition of the aquatic life, become more useful. Aquatic biological indicators are particularly useful because the things that live in water experience all of the cumulative effects of multiple stressors, which you could go out and try to measure all of these different pollutants and stressors individually. But number one, it's not very cost effective. And second of all, you wouldn't be able to evaluate the cumulative effects of multiple stressors interacting on the biology directly. And they also provide an integrated measure of conditions over time. So in the case of, we don't have a lot of toxic pollution in Minnesota anymore, thank goodness, but it's definitely an issue where there could be pulses of a toxic pollutant, for example, that are dramatically affecting the aquatic life in a system. But if you're not there to capture a sample at just the right time, you may be missing, that high concentration of a pollutant. And when you come back, it may look fine, but it's had an effect on the aquatic community. They do provide a reliable indicator of degradation. We've studied this for many years. I'll talk about that in a minute too, but the biology responds in a predictable and understandable fashion to stress. And like I mentioned before, the biology provides this direct measure of whether we're meeting our aquatic life use goals. So talking about, cumulative impacts, this is an adaptation of the DNR's 5-component framework. The biology is affected by everything that's going on in that environment. The hydrology, the geomorphology, this, is more specific to streams. The physical and biological connectivity of systems, and obviously water quality. And this is a link, this is a hyperlink. If the slides are shared, I encourage you to click on this link and explore the DNR's 5-component framework. There's a lot of good material there. So the use of biological indicators in aquatic systems is based on the concepts that some organisms are sensitive to pollution and some organisms are tolerant to pollution. And I work mostly with fish and invertebrates. So for example, brook trout, lake sturgeon, these are the classic examples of fish that are sensitive to environmental pollution. Stoneflies, some types of mayflies and caddisflies. And then we know that other species like bullheads, scuds, a lot of aquatic dipterans or flies, they're more tolerant to environmental pollution. And we can track the relative abundance and presence and absence of these different types of organisms and make some really good inferences about the conditions of that system and what pollutants are impacting the system. You know, a basic concept is that in a natural setting, you have a diverse, balanced community, many species of varying sensitivity to pollution, including species that are highly sensitive to pollution. In rivers, this includes things like red horses, which is this kind of sucker-looking fish here with the red fins, darters, minnows, even things like rock bass, which, you know, a lot of people may not think much of rock bass, but we found them to be actually a pretty reliable, positive indicator of good water quality. And as things become more degraded, those more sensitive species tend to decrease in abundance and may disappear entirely if conditions get bad enough. And they're essentially replaced, you know, numerically by more tolerant species like fathead minnows, bullhead. The green sunfish here is a classic, like, bad actor. Sometimes people talk about how you'll lose different species of sunfish as systems become degraded, and you'll see more and more dominance by this tolerant green sunfish. They're really pretty, but not always a great thing to have a lot of green sunfish in your system. You know, these concepts have been known for many years, you know, going back to European scientists working in, you know, Germany, Austria. They developed this thing called the saprobic system, which was really focused on organic pollution, but they documented how as you get further and further away from the effluent outfall or the source of pollution, they were seeing these co-occurring decreases in dissolved oxygen, increases in biological oxygen demand, ammonia tended to increase right downstream of those sources. And then the organisms that were found in those systems at those same locations changed as the conditions changed further downstream. But this was, again, really focused on organic pollution specifically. And as time went on, more modern biological indices were developed, like the Hilsenhoff Index, which also focuses primarily on organic pollution. But it was a more sophisticated system looking at a wider range of organisms, a more community-based assessment instead of just a couple specific biological organisms. And it had like a scoring and rating system associated with it. And then in the 1980s and moving onward from there, we've moved into this concept known as the Index of Biological Integrity. This is a more comprehensive tool looking at multiple aspects of biological communities. We call them metrics. So this could be, in terms of the fish community, we might look at the number of piscivorous species. We might look at the number and abundance of species that reproduce in different ways, like they might need a certain type of substrate to spawn. Just their basic pollution tolerance or sensitivity. It's using different metrics of that biological community to evaluate the overall effects of human actions on these living systems. And I think Jacquelyn will have some examples here, but these are chosen, the metrics in each IBI were selected in an empirical fashion, looking at the response of the community to either a specific stressor or a more a gradient of combined stressors at times. But they're known to respond in a predictable manner to that stress. And yeah, we do collect a lot of other data associated with the biological data. Habitat water chemistry, hydrology, and so forth. The IBI tools we use in Minnesota, in streams, we use fish and aquatic macroinvertebrates. In lakes, we use a fish-based tool. And we also have tools for evaluating the condition of wetlands. We don't have a wetland scientist on the call today, but if folks are interested, I can certainly refer you to them. In those systems, we use aquatic plants and macroinvertebrates. Okay, so with that, I think I'll pass it over to Jacquelyn to talk in more detail about the lake tools. Can you request control, Jacquelyn, or would you like me to advance slides?
I requested control, so see if you get a.
Okay.
All right, I think I have it. Yeah, so I already introduced myself. I'm with the Fish IBI program with DNR. And as John alluded to, you know, we developed, or we have a lot of diverse types of lakes in Minnesota and a diverse landscape. So we work statewide on all types of lakes that are experiencing all sorts of different types of stressors. Let's see. I don't think I'm actually able to advance the slides, John.
Oh, well, let me see if I can.
Yeah, see if you can.
Okay, it's not working for,
Oh, okay. So I think you might still have control. I'm not sure. So, and a lot of what I'm mentioning, you know, we're using lakes as a specific example, but there's very like similar types of rigorous study design with the stream IBIs that John works with. So with the lakes, we use 4 different sampling gears during the summer months, and the map on the left just kind of shows the different colors or different types of sampling gears that we're using, and shows the distribution that we are sampling, you know, all the different types of habitat within a lake. We've developed tools for lakes from 100 acres to 10,000 acres, more or less. we flex that a little bit. Yeah, I think you got to advance it, John.
oh, okay.
Okay, so John mentioned this, that when we're selecting metrics for the different IBIs that were developed, we look at relationships with the physical habitat in the lakes. And so John talked about this early on in the call for those of you that were on, that, you know, if you have complex habitat, you have woody structure, if you have different types of substrate, complex vegetative communities, we see a very different type of fish community than we do where we have, you know, a lack of that habitat or just a very homogeneous habitat. And then similarly, we see fish communities change in a pretty predictable way with changes in water quality associated with sedimentation, algae, and then particularly some of the cold water species that are sensitive to deep water temperature and oxygen requirements. And so the stressors that, you know, I think everybody's kind of familiar with are primarily what's going on in the shoreline, what's going on in the watershed, water level. I'll talk a little bit later about AIS as well. Next slide, John. So we didn't get brought into the project until a little bit later. So John kind of showed the stream, how with the streams, they have actually completed the entire state. We still have a couple of years left. PCA asked DNR to help with assessment of lakes. In about 2011 is when we kind of started up our program. So the map on the left just shows the percentage of lakes that have been assessed within each of the watersheds that have been kind of fully supporting or in good health based on the fish IBI. So the green watersheds, you know, 3/4 or more are in good health. The yellow, it's over half, and then the orange, it's under half, and the red, it's under 1/4. Or in some cases, all the lakes in a watershed that we've assessed have been found to be impaired based on the fish community. And then on the right side of the map, We also look at exceptional lakes. And the blue on the top would be where over 40% of the lakes that were assessed were found to be exceptional, and then lesser numbers. And you kind of noticed in the agricultural areas of the state, there's very few lakes that seem to retain the fish communities that would have been probably pre-settlement diversity. Next slide. Okay, so you can kind of click through this one, John, because I think it's animated. But in general, John talked about talking to other states. I've been really fortunate. We were one of the first states to be assessing lakes, and so I've been in communication with a lot of lakes. And I think we forget living in Minnesota just how fortunate we are that in a lot of ways our lakes and our fish communities are generally healthy. But we definitely see issues, particularly in agricultural and urban areas, and most often the lakes that are impaired for the fish community are also nutrient impaired. And then most often the exceptional lakes, nearly all of them are in forested areas of the state, have diverse plant communities, have high quality shoreline habitat. They're really of different character. So we look at stressors that are impacting lakes as part of our fish IBI team. That's part of our responsibilities as well. And eutrophication and physical habitat alteration, primarily shoreline development, are the most common stressors that have been identified. I think there's one more bullet. But it's not to discount that John talked about kind of the cumulative effects. And so there's non-native species. Connectivity can be incredibly important. And then there's a lot of stressors that are kind of difficult to measure, such as impacts from pesticides. John talked about the kind of ephemeral, the very limited monitoring data, and you almost can never catch if there is an event. And then we have been doing some work looking at cold water communities and the response to climate change, but again, that's a pretty complex issue. So this just shows 4 graphics looking at the fish IBI assessments. So whether a lake has been identified as having an exceptional fish community, which would be the very highest of scores, fully supporting where it's not impaired, but it's not quite as high of a fish IBI score, or it's not supporting where the fish community has pretty much lost nearly all of the sensitive species, and you start to see some shifts in composition. So instead of having a lot of bluegill and sunfish, maybe you're seeing more bullheads and carp and such like that. So the purple are the exceptional, blue are fully supporting, and the red are not supporting. And so you can see in all cases, we have a pretty strong relationship with total phosphorus that's in the system, with watershed disturbance, with the shoreline habitat quality. That's measured with in this case, I'm showing score the shore scores, but we also look at dock density, other types of shoreline measures, and then with aquatic plant richness. So to get at some of those relationships with the shoreline, we do a pretty wide variety of habitat surveys. And I know that some local watershed districts are starting to get interested in collecting this, and some have been. Some consultants have been collecting this data. And one of the efforts that DNR we have going on right now is to try to make some of the tools that we've developed for habitat surveys, get them out from behind our firewall, because there's a lot of interest in other folks collecting them, and they're pretty simple tools where with someone who has some savvy and can read a manual, can probably pull off some of this. So on the left-hand side, that map is showing 2 different types of surveys on the same map, but so we've done a plant community mapping of floating leaf and emergent vegetation. That's the polygons, and then the little star circle shapes are showing what's called a score the shore habitat evaluation, looking at what is the character of that habitat. And then on the right hand side are two different types of surveys. In the upper right, it's looking at using a hydroacoustic evaluation of the plant community. So this would, in addition to would include submerged vegetation. And then in the bottom right, looking at just a shoreline classification. Is it altered or is it natural? So really simplistic that most anyone could do. And then just an example that kind of maybe help folks kind of wrap their head around. And John and I, if we would have gotten to work on this a little earlier, we were talking about, we could have had one with streams and lakes, but in this case, we're just using lakes as an example. So the Crow Wing River watershed was a watershed that we assessed this past year. And in this case, I didn't add up the number of lakes, but it looks like about 70-ish lakes where, we had huge, we don't have very many watersheds like this, where a really big proportion of the watersheds are proposed to be protected at an exceptional use designation level. So in that case, you're protecting the water quality standards that are being proposed are more stringent than for other lakes. And a lake has demonstrated its capability of supporting a fish community, having the habitat there. And then we have, a handful of lakes that are fully supporting, a couple of lakes that are not supporting. In this case, there's just one lake that was identified as vulnerable to impairment. That's another set of lakes that is where local units of government and other planners, we're hoping that those vulnerable lakes would get extra consideration for protection. Next slide. Okay, I think, you can pop through these ones too. So IBI information, whether it's stream, wetland, lake, IBI, it's one piece of the puzzle to be used for prioritization. And it kind of depends at what level somebody's looking at. Is it a local unit of government that is working on shoreline standards, or is it a watershed level where they're looking at, which lakes within a watershed, which streams, which wetlands, are you going to get your biggest benefits with? And so there are a variety of tools that have been developed, but there's also just simply a lot of information out there. And I'll talk about where to find some of that information coming up here. So vulnerable and impaired lakes and streams are also investigated and have pretty thorough reports that are put together. So for the lakes, that's my unit, the DNR, that does that. And then for the streams, the stressor identification is done by PCA staff within their watershed unit. And exceptional lakes are out there, and then as well as cold water streams that support cold water fish and bugs, and lakes that support cold water fish that we know are more sensitive to eutrophication, but also to climate change, and so should be protected or prioritized for protection. Next slide. Okay, John and I sort of struggled with this because I think one of the main questions we get when we talk to local governments or consultants, or somebody who's not a DNR or a PCA person, and even DNR and PCA people, is like, where do I find all this information? And it's actually it's hard because it's spread out. So I've listed, and I'm hoping that you guys are going to share this presentation afterwards too, like where you might find some of this. But you can always just contact John or me as a starting point. We can point you in the right direction as well. So there's GIS level data on MinGeo. We have most all of our DNR data on there. PCA has this water quality assessments results viewer. And that's what's shown in this graphic here where you can get a lot of information, mostly PCA collected information, but some of the DNR stuff is in there too. DNR has a watershed health assessment framework. It's called a WAF that also has a lot of PCA and DNR information. And I personally find that to be probably the most user-friendly. If somebody doesn't have a lot of experience with GIS, you're just clicking layers on and off on a browser. PCA, if you're looking for extensive water, like the stressor identification reports, detailed information on assessment and monitoring, that would be on the PCA watershed pages. And then if you're looking for raw fish survey data, in our case, that'd be on Lake Finder. In PCA's case, some of that would be, John, you correct me if I'm wrong, but I think you can get some of that with this water quality results data viewer. I think you can get down to the raw fish data. And then John also mentioned the long-term monitoring of rivers and streams, which is of particular interest. And I put the two, there's a lot more websites on this, but I put the two websites that are directly for IBI. And like in our case, if you go to the Lake IBI, we would link to pretty much all or most of those other pages that I talk about. We try to make it as easy for someone to find what they need to find. Let's see. And then next slide. Oh, so I briefly mentioned stressor identification reports, but I want to just reiterate that those are on the watershed, PCA's watershed website. Although there is a delay, I think we have like 6 or 7 that are waiting to get published for our lakes. So you can also just reach out to John or I in the meantime. And we've got 24 of the watersheds completed for lakes. Like I mentioned, we started later, and I think nearly all the watersheds are completed for streams. And then I just wanted to give an example. So for one of the products that we were asked to produce, a couple different watershed districts, and folks that are like, hey, can you just help me out here? I wanna, I don't want to read, through all these different documents. So one thing that we've done now is every lake that we assess, we do a 1-pager. And so this is an example of Bacigalupi Lake in Pine City, Minnesota, and just kind of goes through what's the fish community like, what are the stressors, what's our recommendations. It's a 1-page handout that in some cases, it's always included in the stressor ID, and then in some cases, some of the authors of the RAPs have included them. Next slide. Okay, John, do you want to talk about this?
Sure. I'm not muted, am I?
Yeah, we can hear you.
Okay. So to just take a step back from the really granular stuff that Jacquelyn just directed folks to take a look at, I wanted to mention that these tools are also being used at a statewide scale to track progress towards some of the real big picture goals that we have, our big picture long range goals. So in this Clean Water Roadmap, which was first put out back in 2014, but it's being updated every couple of years, we had this statewide goal of by 2034, we want to increase the percentage of rivers and streams in Minnesota that have healthy fish communities as measured by this IBI. And so this is a way that, we track progress at various scales, but this is one way that our IBI tools are being used to kind of take a step back and just evaluate big picture how we're doing. And so far, go back, the progress, it hasn't been dramatic. I think we're kind of holding steady, maybe with some small improvements kind of at this statewide scale, but I think that's understandable given some of the challenges we're dealing with. And yeah, I just wanted to make sure that folks know that this is not just a fine-grained tool, that we are kind of trying to roll this up into some broader summary. All right, I'll pass it back to Jacquelyn.
Oh, I think that's all I had, and I just listed. So our fish IBI staffers, we're a fairly small crew, so this is us. And I listed the different watersheds that, you know, who would be currently the contacts for each of the watersheds. And our website, again, is up there. You can also just, it's not easy, you know, like that's a crazy address, but if you just Google Fish IBI, Minnesota Lakes, or same thing, I checked John's. John's comes up with Stream IBI, so.
Yeah, I don't have mug shots of all our staff, but that's something I can work on for the next talk.
These are pretty old. I think most of these are a few years old. That's all we had.
Thanks, John. Thanks, Jacquelyn. We do have a few questions from Anne I think we can get into here. Anne, I don't know if you want to unmute yourself and ask them yourself. If you don't unmute, then I can go through them.
Sure, I will go through them.
Okay.
Back in the day, like early 2000s, I did some IBI invertebrate sampling on wetlands, and we ended up like stop collecting it because it just seemed like there was way too many errors and like things that maybe the invertebrates were there, but maybe your trap was set poorly or whatever. But this was a long time ago. So with invertebrates, do you feel like that's still kind of an issue, or are you finding better methods to sample them? Fish, it's easier. I assume that was more shocking. You can find they're there, you know. But with the invertebrates, they're kind of tricky. So do you have anything to say about that?
Yeah, I'm not sure if you were using like emergence traps or something.
I feel like there was some kind of bottle trap that,
Okay.
yeah.
Yeah, so our methods for invertebrates, both in streams and in wetlands where we have those IBI tools, are they're direct sampling techniques. So we have the biologist in the stream, in the wetland, manipulating the different habitats to collect invertebrates off of them. So in streams, we use D-frame nets, and we will target different types of habitat. The four core scale habitat types we target are rock, wood, submerged macrophytes, and then bank habitats, which can either be undercut banks or overhanging vegetation. So the biologist will actually physically pick up a rock or take a piece of wood and dislodge the organisms off of that habitat with their hands.
Cool. Glad to hear it.
Our standard approach is to do 20 what we call efforts. So like 20 1 square foot surface area samples in a sampling reach. And we put all those 20 samples into the same bucket, and then there's a subsample process to pick out, I think we do a 300 organism subsample pick. And we found that we found pretty good repeatability with that. We don't feel like we're missing a lot of productive invertebrate habitat or not capturing organisms that are there in the stream or wetland.
No, that definitely sounds way better than what we were doing. So glad to hear it. I feel like maybe we have programs in the metro here, like the WEP program, and I think they just do the bottle traps. And so I don't really, you know, so therefore the data they get is kind of like, I don't know, but glad to hear that it is that intense for this sampling you're doing.
Yeah, it typically will take a biologist anywhere from 45 minutes to sometimes an hour and a half to, you know, sample a stream.
Yeah, it's a lot of work.
Yeah, they do it thoroughly. And
looks like Jill might have a related question.
Let's see. What's the question?
You're on mute, Jill.
Thanks. So being part of a watershed district, we do a lot of monitoring, and we do an annual report on fact sheets on each one of our lakes and our 3 creeks. The problem, you're talking about making progress. It just seems like we need more education for the citizens of the state, because if there are some bad practices, that can influence what's going to happen in the water bodies. Are you seeing any of that? I mean, is it dependent on the practices within a region, or and how much value is putting on their conservation efforts? Does that make sense?
Yeah, I think, John, you can hop in too, but I mean, I actually in previous jobs have had more engagement with that, you know, with working directly with community education and outreach. You know, we do, this kind of relates to Anne's question too about do we share our results? So, you know, as now kind of like managing biological data and assessment, you know, we have kind of a limited, we do share our data through kind of the within RAPS and One Watershed, One Plan, and through, you know, these different planning exercises. But it's not our units as much out there doing the public education. That's at least in DNR, it's, you know, our outreach unit that is primarily working on that. You know, having said that, you know, I'm always, maybe I am a fairly optimistic person. So, you know, I think, was it Kristen, like at the beginning, you're talking about like pollinator, you know, we see like how the pollinator stuff is kind of like taking off, where people are getting, all of a sudden it's the new thing. Within some of our fish IBI work, some of the work that we've been doing with cold water, I didn't talk about it in this presentation, but cold water fish communities seems to be getting a lot of press and people care, and all of a sudden, you know, there's a lot of interest in protecting them. So I do think there is, you know, there's these public movements that happen that as DNR and PCA, you know, we need to kind of capitalize on them, but we're probably not the experts. I don't know, John, if you have something else to add.
Yeah, I'd say a couple things. You know, I'd also point to the buffer law as an example of something that was driven, at least in part, by a recognition of how landscapes interact with aquatic systems. And obviously, you know, stream buffers provide a variety of other benefits besides to water quality and biological health, but I think it was a very intuitive connection for people that the land immediately adjacent to water is of critical importance to that water, and we need to take steps to protect it. And I know it wasn't universally popular. Like I mentioned, I work in the Red River Basin, and I run into landowners all the time who feel like they were, you know, kind of coerced into taking land out of production to protect a water system that they don't have a lot of interest in, you know, seeing maintained as a resource for aquatic life. They just think of it as a ditch, you know. But I think in general it was a broadly popular initiative. So I think that's a little encouraging. And then there are other folks at our agencies who work more directly on the outreach, like Jacquelyn mentioned. But local partners, you know, they are the ones who are empowered to implement these changes. And I agree that education is a critical part of that, but there's also just the need for buy-in, you know, for folks to say, yes, I care that this water system is polluted, or it's a high quality water system and I want to do things to protect it. And really, until, you know, people feel that, you know, fundamentally it's always going to be a challenge to change those practices.
Do you feel like, John, that the buffers are doing their job, or is there enough diversity within the buffers to really create good soil systems too that support?
Well, you know, that's something that it's probably going to take some time for the effects to really be manifested. I think that is something that we will be trying to suss out over time as we revisit watersheds where buffer implementation has been widespread. We're hoping that we do see those positive impacts on habitat and water quality, but I think it's been a relatively recent development that sort of documentation of buffer conditions and then efforts to, get them put in where they needed to be. That's relatively recent. I wouldn't really expect there to be a, immediate response. But over time, it should have a positive impact on water quality. And some of these things are gonna, it took us a long time to get to where we are today in terms of, the effects of pollution and watershed degradation, and it's not going to be a quick 180 turnaround. I don't think that's necessarily a realistic way to look at it. I think it's a longer term goal, and documenting progress is important, but we really can't expect there to be a silver bullet right now.
Okay. Well, I represent watershed districts on the Bowser board. So I see the RAPs, the importance of RAPs to planning efforts, and especially with the one watershed, one plan. So I keep asking, so is there a lot of diversity in these buffers, or are they just a monoculture? But I never get an answer. So I was just curious as to how we're doing here. So thank you. You guys are doing the important work of collecting the data to inform the decision makers. So this is really good information. I'll probably be reaching out to both of you to learn more. So
yeah, the other thing I was going to say, I keep coming back to the Red River Basin. I know that a lot of folks on this call might not be that interested in ditches, but that's one place where I see a lot of opportunity for restoring watershed function. It's an example of a situation where we have somewhat competing interests in, drainage. Agricultural producers need agricultural drainage. And ditches are parts of watersheds. And so trying to find a way to achieve our drainage objectives while still protecting aquatic habitat and water quality, I think there's a lot of opportunity there, not just in terms of buffers, but in how we construct and manage our water systems, providing floodplains, building sinuosity into channels to, allow for sediment transport to become more natural, nutrient sequestration. That's an example of an active process out on the landscape that's just ongoing, where we can do things in a better way that will have a positive impact. Anyway, that's kind of a tangent, but thought I'd throw it out there.
no, I appreciate that, because I've been thinking along those lines. How can we improve that? And being in an urban area, I mean, our storm sewer systems are equivalent to your drainage ditches, only we're not doing a whole lot, seems like. So I'm anxious to hear from Jack too.
Okay.
Good point, Jill. Couple more questions here before we move on to Jack, I think. So another one from Anne was, so John, Jacquelyn, are there any conversations happening about pulling all of this data? You showed those couple of slides of here's where you go to find this, to pulling all that together in one more, user-friendly kind of mapping tool or data source. Is there, any plans for some better user-friendly tools?
Well, yeah, I think we're getting better at some of the data visualization tools, but there is a lot of different types of data out there, so it really depends on what exactly you're asking for. In my work, we focus a lot on assessments, and I don't know if people are interested in assessment level data versus sort of parameter level sample information. So there's kind of what level of detail are folks interested in? I get people just saying, send me all the data, and that's a little bit challenging because
Sure.
I'm not really sure what they're trying to accomplish.
Yeah,
Okay.
so maybe better to reach out with those, specific questions that you're trying to do. And we like the one-page fact sheet. Very helpful too.
Yes,
There's another resource too, and that's your watershed districts. You talk about the ones in the Red River Valley. They're really active. There's a lot of watershed districts up there area. But and then we've got soil and water conservation districts. So those are the people that take the data and try to develop plans and figure out how they're gonna improve things. So that's another place to reach out and see how the data is being used too, because they do education and they may be more supportive of what your concern is.
it just seems to me like a web-based map system where, hey, you're looking for information on this lake. You go to that lake, you're able to click it, and then you might get a list of all the things you can possibly look at. It's, I don't know, just a way we keep trying to go instead of, just searching for text in various locations. It's very hard to find all these things, even if you give a direct link. But I just want to say too, in general, with the DNR, we typically don't have any idea what you guys are doing potentially on lakes in our city. And it's just like we're finding, oh, apparently they did a fish survey and we didn't even know about it and stuff like that. Then that was my other comment on, would just love when DNR works in a certain area that they're able to reach out to the communities or the watersheds that they're in and making sure they know what's happening and what they're finding. So thank you, though. It was all really great.
Okay, one question here from Mars too. I don't know, John, Jacquelyn, if you can speak to any further about some of the water chemistry quality testing that I'm guessing some of your colleagues do.
Yeah, I guess I can talk for lakes. It is completed by PCA and by grants that they give to local partners. So within the watershed schedule, so John kind of showed how we're marching around the different watersheds. Typically, local stakeholders and partners, and even DNR would request of PCA, hey, we are interested in collecting water chemistry data on these lakes or these streams that are coming up for assessment. And that would typically include phosphorus, chlorophyll A, Secchi readings, and then in addition, temperature, oxygen profiles for lakes. So that's all completed within that same framework, that same schedule of, moving around the watersheds. And there's some prioritization. The first round of assessment, they were looking at, kind of trying to get the broad picture. What's the state of, I think they tried to get to a pretty large number of lakes. And with this, now that PCA is moving into watersheds for a second time, it's some of that getting an overview, but it's also some of the targeted monitoring associated with, okay, we know projects were done by watershed districts or by whoever in that might impact these lakes or streams. So we want to do additional monitoring to see if we see changes, if things stay the same, et cetera. And so so there's opportunities for input on which lakes and streams and, are being monitored, whether it's for water chemistry or biological. We take all that information. I think in our case, we almost are always able to accommodate any requests that we make for fish IBI or that are made for fish IBI. I think there's a little bit more demand for water chemistry, so they have to prioritize. But John, if you want to add anything.
Yeah, for water chemistry monitoring, the PCA does have the watershed pollutant load monitoring program too, which does a lot more intensive water chemistry monitoring. They're out there chasing snowmelt events. They're looking at sampling after storm events to try to track the quantity of pollutants that are being transported through our watersheds. They're not so much focused on kind of ambient conditions. They're really doing intensive sampling to monitor the flow of the loads of pollutants that are coming through. So that's another aspect of our water monitoring program that we didn't really touch on today. It's really a separate piece that's related specifically to water chemistry.
Thanks, that's helpful. All right, I think we're ready to switch things over and hear from Jack. So John, I think you're still sharing your screen if you would. And Jack's gonna share.
Yes, I'll share.
Too? Okay.
And I'll have that ready. So yeah, let me just get this prepared up. So yeah, hello everyone. My name is Jack Distel. I am the Water Resources Specialist for the City of Bloomington. And Bloomington is a part of the GreenStep Cities program, so I'm happy to present to you all today. Similar to like most places in the state, Bloomington has a lot of water, even after the chaotic filling and bulldozing that happened in the mid 1900s. And the vast majority of these are small ponds, and they're connected to our municipal stormwater system. So they're not, I mean, an engineer might say they're not stormwater ponds because they're not designed and constructed, and they're regulated under WACA, the Wetland Conservation Act, but colloquially I'll call them stormwater ponds and lakes just because that's a driving system characteristic of them. And so Bloomington does have a few healthy waters. If you have been to Bloomington, Bush Lake is our gem. It doesn't have very much stormwater input at all, and there's a few other ones scattered around. But being that this is an urban area, a lot of our waters aren't doing too well. We have 2 impaired waters, listed impaired waters, and 3 if you include 9 Mile Creek, which ends in Bloomington as it exits into the Minnesota River. And all those little ponds aren't doing so great. Even some of the ones that aren't connected to the stormwater system, mainly because of, poor riparian habitat and whatnot. So my job is really to try and understand how we could change our practices to improve water quality across the city. And I'm much more focused down than what Jacquelyn and John presented on because I have a much smaller land area. But we do a lot of sampling ourselves. And that includes, we go out in a little raft from April to August, and we get grab samples once a month on about 30 water bodies, and we'll kind of shift that list over time. We have some ones that we go to every single year, and then we'll have like 6 or 7 that sort of change over 5-year periods just to get a good distribution across the city. We have data going back to 2009, and we're collecting things as you would collect, chloride, chlorophyll A, conductivity, phosphorus, dissolved oxygen, temperature profiles. And it gives us really interesting data. Mostly we can see that we're having a pretty big impact on our water quality. But we're also doing some additional grab samples, looking at some biological characteristics. And so, we take a percentage absolute coverage of floating leaf dominant, whether it's duckweed or lily pads or filamentous algae. We get a transect survey using our viewing scope for submerged aquatic veg, and we get an absolute coverage there, so we can get just a really quick snapshot sense of which water bodies are dominated by submerged veg, or they don't have any submerged veg, or they're kind of stuck in the middle where they're starting to either gain or lose submerged vegetation. And we look at sheltering, so how much tree coverage is around the edge of these ponds. When you look back in the historic aerials, a lot of these little wetlands wouldn't have had such dense forests around them, and we've let that grow up over time. So that's really affected a lot of the system chemistry and how they react to the system itself. So if you're like me, a water manager in an urban landscape, IBIs are great and they sound great, but the big elephant in the room is that I can confidently guess how most of our waters will score on any particular IBI. Not well. I think we were in with the map Jacquelyn showed in the gray area that shows that we have no well-performing waters. So a big culprit of that obviously is the urban ecosystem for us and agriculture for agricultural systems, but in an urban ecosystem, I'm sure cities they're watching this now are going to watch this in the future. You have a map very similar to one being shown, which gives an example of the big culprit for how these systems function in our stormwater network, which covers the entire city and it moves water fast and efficiently, and that increases the transport potential of all those pollutants from point A to point B. And point B tends to be these little water bodies. And everything that lives in them is affected from that as we go on. As an ecologist, I'm not an engineer, I try to redefine these stormwater systems as urban subterranean streams because they are a natural part of a urban ecosystem. A beaver dam's natural out in the wilds of the boundary waters, and these are what these novel ecosystems have. And so thinking about them like that as a novel ecosystem type that we're trying to manage for, that we don't really have good reference sites to compare to, is an interesting way to look at it, and has helped us start to create good benchmarks for management in the long term. So as Jacquelyn and John pointed out, IBIs are really good for putting together a program of protection. So, where are the high quality sites, where are the low quality sites, and they're really good also just like put together a list of prioritization. So if we know that one particular water body is a big main culprit and it's kind of a key part of a particular watershed, then we could say, well, we want to put our money towards, improving that water body. Or if it's on the verge of being pretty good, then you can start to try to prevent it from, degrading further, and then maybe bump it up into a high quality system. In Bloomington, most of the damage has already been done, so we're really working with mostly degraded water bodies. And so we do have a prioritization schedule. I mean, the impaired waters list really helps with that. So like we'll say, well, Penn Lake, which is kind of in the center here. I don't know if you can see my mouse. That's lower Penn Lake. There's an upper lobe to that. That's one of our impaired waters. So we'll say, well, that's a priority to work on. It's impaired. We know that it's in bad shape. And but biological indicators are still very helpful for that. What I'm going to talk about in this next bit isn't really about how biological indicators work or what we're using them, how to measure them, because I'm not going to do anything better than what Jacquelyn and John talked about. I'm going to talk about for managers how we can incorporate those IBIs into our management plan and goals, to try to see not exactly how these systems are reacting to historic stressors, but how they can be used to perceive and assess and help plan for reaction to stressors that we're putting in specifically to improve the system or improve our management. And a great way to do this is, and I recommend everyone to try to do it when you're starting to work through everything, is creating ecological frameworks. So basically, this is how systems function. So when stressors have been tossed around a lot in this presentation, so basically, ecological frameworks show where those stressors lie in it. The first line is usually drivers of the system. Those are big, high level characteristics that are really difficult to change, climate, rainfall amount that goes over the landscape. The example on the screen here is for a shallow lake. And so, groundwater that flows into it and through it, you got the storm water that goes chugging down the roads and outletting into the lake, and then you have a little bit of overland flow. Those are going to really drive the system itself. And you can make these as complicated or simple as possible. This one's rather simple, obviously, but makes it easier to read for a PowerPoint slide. And then, then you have stressors, characteristics, and system attributes. So the stressors are really what's going to be defining a lot of the internal system characteristics. So, water clarity or turbidity, the nutrient level in the water, and the water chemistry itself, and how those stressors are, you know, shown in the system itself. It will dictate how much submerged aquatic vegetation you have. If the water is really turbid and it's going to block sunlight, you're probably not going to have anything growing on the bottom, and it's just going to create these feedback loops. And then that starts to create these fish species composition. So those system attributes that here at the bottom, that's where those IBIs that everyone are talking about come into play. So you're looking at those to try and see what exactly are these stressors um, doing to the system. And that's really helpful from a management standpoint, because you can start to say, well, we have this idea of what's stressing it, and we can start to identify where we can fit ourselves in within this whole framework to start to tweak the system. So, and you're looking at a urban shallow lake like this, you got storm sewers. Well, we already do a lot of infrastructure interventions. So we can say, well, we know we need to change the trajectory of stormwater flows hitting the lake. We're not going to get rid of stormwater sewers. It's just not feasible. So it's a natural part of the system. So how will they act in a more sustainable way in the long-term future? You're starting to reduce that transport potential of that water as it moves through the system. So, street sweeping, reducing the amount of salt you put on the road. That's a source prevention. You have rain gardens and little bioinfiltration basins scattered across the landscape that helps soak things in, that slows the water down before it gets to the storm sewer. So it's, also stopping some of that transport potential. Then you have some of the more expensive infrastructure interventions. You can do traps, you can do different ways to filter the water as it comes out, so on and so forth. And then you have in-lake or in-system management. So with those IBIs, when you go through and you know green sunfish, carp, black bullheads, they're just dominated this turbid little shallow lake. That's good to know. One, it gives you really good data to assess in the future. So, how often you're measuring these things is always changing. We do water quality often because it's very variable. It changes a lot. These biological systems are a little bit more slow moving. They show a longer term trend, but they can also create internal feedback loops that self-support their own existence. So when you have internal loading and turbidity being stressors, and you have fish species composition being dominated by carp, which are rooting around in the bottom, they knock up this total suspended solids, which pushes up the turbidity, which decreases the macrophyte distribution, which helps the carp dominate the system and not have any of those bluegill predatory fish that might eat their young. They have a self-sustaining feedback loop. So you can start to mess with the system both from the high level drivers and stressors, and then also use interventions at the bottom level to stop these feedback loops from continuing onwards. So you're trying to transform the system into something different. So whether or not you can flip a shallow lake to clear water state, I don't know. It's all dependent on each specific site and zone. So when you do, as a city, you're always going to be challenged with, money and how much it costs to do these assessments. We don't have the staff time to go and do IBI sampling. I would love to in the future potentially do um, macroinvertebrate sampling. If we could, I think we could maybe do that in-house if we could figure out some way to do it. We'd have to get some new equipment and everything, but generally we contract it out. And we'll contract out wetland inventories across the city. We'll do that like every 10 years. We did, MinRAM is kind of a type of IBI. It's, assessing high, low, middle quality wetlands, and then we can put that in a way to figure out which ones we want to go after first or protect and whatnot. In this shallow lake restoration plan, we just did this huge full year, pretty intensive biotic investigation. So looking at all the different parts of the system, that gives us baseline data. Yeah, we can kind of guess what kind of fish are in there. I go out there often. I kind of know what's in there, but we also found some mussels living in this it's illicit impaired water, and there is a mussel population living in this lake. So there are sometimes stragglers that hang on that can show you what the system is going to be. So Penn Lake, this is the conceptual model for Penn Lake. It's never going to be what it used to be, or it's not going to be a shallow lake without it. You can't make it a reference shallow lake that doesn't have stormwater inputs. So the IBIs also help you identify maybe if these fish communities, macroinvertebrate communities are living there, you can start to tweak it like, okay, what the things those animals need to live. Well, this more preferable creature, what it fish, macroinvertebrate bird, whatever it is, kind of also lives in a similar system. Maybe we could tweak it and push it towards that way. And that's that novel, rehabilitation strategy of managing it. And once you start to do that, and you have those IBIs in there, you can start to do scenario forecasting. And so, easy thing to do. So you make a y-x-axis and you put how the system's going to change over time. So say, for example, on the y-axis, you have sustained water quality at the top, degrading water quality at the bottom, turbid state going on the x-axis to the left, and clear water state on the right. So, if you sustain high water quality, if we're able to like knock down with our street sweeping program, better infrastructure across the watershed, so that stormwater input that's going through is much improved, then you can kind of project what's going to happen in the system, and you do like carp management and internal stuff. So rough fish population low and management minimal, self-sustaining game fish populations, high diversity submerged aquatic veg. On the other hand, you could say, okay, well, if we do all this intervention, we do a big alum dosing across the lake, and it's like 1 year of really good water quality, but it just starts to degrade from there on out. Well, then you can start to say, but it goes to turbid, so the rough fish population starts to come back in, and low diversity SAV. So it can kind of give you a sense of where your priorities lie. So we know that whatever we need to do, it needs to be sustainable. So that water quality, that driver needs to be there. And then this helps communicate to the public what might happen. There are a lot of cases where, you have your degraded shallow water lake, and it's turbid, and there's a lot of carp in there, and you say all the indicators show that this thing is not doing well, and say you successfully flip it to a clear water state, and the submerged veg take off, and all of a sudden you have matting on the surface, and everyone's like, oh, I hate plants, you've ruined my lake. So this also helps show a little bit to the public what might happen and how these systems react. They're their own, unique things of the community. They're not just a resource or a commodity, and so they're going to shift and change. We got to kind of adjust our expectations with that as well. So I particularly like that for it. What else do I got here? So ultimately, with any of these restoration plans, the main goal is, you're going to take your biological indicator measurement, whether it's, from a city point of view, we'll do those big ones, like I mentioned, MinRAM, and you select, protect, restore, you have your prioritization plan. Once you get that, you go to a whole different scale of investigation. So you look at this one particular water body, you're going to do a restoration plan for that and improve management. Well, you got the whole driving watershed scale. You take that management ecological framework, and you're using those IBIs to kind of drive what is possible, what's the goal, and how you're going to interact with it. So in 10 years, we'll go back through and we'll also look at those indicators as well. It gives us an ability to set SMART goals. We can say, we want to reduce the carp population by this percent by this much. Well, we had to get that indicator to even set those SMART goals. So it does give you management criteria that you can meet. And you really only have to do it once in a while if you're also including other measurements that can kind of identify where the system lies. So water quality measurements being the common one. That's pretty easy to do. The infrastructure is there. It's pretty cheap. So you kind of fit that in a reoccurring maintenance mechanisms, and then you put in these IBIs over time, you can really set forward a, clear story of where that water body is going, which is a bit different than like a lot of like what the state's doing, is like getting a clear picture of where the water has been and where it is right now. And so from a city point of view, since we're the active managers of those systems, I'm trying to say why this is going to be a really useful tool for you all to, set forward and get a clear path going forward. So that's really all I had.
Thank you, Jack, for sharing that city
So
perspective. And
I'm sharing my screen, huh?
yeah. Oh, but you have a cool turtle background.
Yeah.
No, I find that really interesting, and thank you for making that connection to how IBI is, useful in terms of goal setting and plans, which, is very much a local government um, activity. And so I think that that's really helpful to make that connection. So I don't see any questions in the chat, but I want to open up for anyone else. Otherwise, I have one.
I wanted to make a comment, but you
Yeah.
go ahead, Kristen.
Okay. Well, I was wondering, so you mentioned that you typically contract out some of the IBI testing in the community. Are there, and maybe John and Jacquelyn can answer this too. Are there opportunities for volunteers to do any of this? Community members, students. There's often like citizen science. We talked about pollinator habitat earlier and how easy it is to go out and take count of what butterflies you're seeing. Are there similar opportunities for community members that you know of?
The wet program's ended right now, so that one's kind of done for as far as I've heard going into the future. Maybe I'll start back up. That was a big one that was slightly useful. I mean, I agree with, I don't remember who was saying it, but kind of take that data with a bit of a grain of salt. I think when you do things like macro inverts and things, there is a bit of talent involved in that comes into play. But you got like the Great Backyard Bird Count put on by the Audubon Society. You have water quality tends to be something that's pretty easy. So there's 9 Mile Creek Watershed District has a really good salts one. So they're tracking road salt pollution, and that is helping us identify some hot spots. We're trying to decrease our use of road salts, or if we can identify where they're going out in big proportions, we can start to like tweak maybe what we do, or ask the county to tweak what they do. It's difficult for us to set up the volunteer opportunities. They tend to be very staff intensive. So we rely with WEP, we relied on Hennepin County to do that. Obviously that salt count, that's 9 Mile Creek Watershed District. I'm set up a community group for this particular watershed or lake restoration plan. And in that, we've already identified a want for community science. It's just we're going to have to talk it out and see what's reasonable and what's useful to us. So I do think birds tend to be one that is pretty prevalent, or amphibian calls, or things like backyard naturalism can really help you do. I think once you start getting into like speech ID, or where you like need more labs to do that, it becomes a little bit trickier, because you're like, well, what is it worth it? And are we just getting that data, saying thank you and putting it in our files and never looking at it again because we don't really trust it? I don't really want to do that to people, just give them a false sense of interaction. So it's turned out to be trickier to get volunteer help than I would have thought before, but we are trying to figure things out like that.
Yeah, that's a really, gosh, even like I have a natural resources management degree and I can't go identifying fish species. Like,
Yeah.
like, no, don't trust that.
Yeah, I'll jump in quick. This is something that we've been thinking about quite a bit at PCA. We do have a volunteer monitoring program for water chemistry, but we haven't yet extended that concept out to the biomonitoring side of things, but it is something we're working on. I think like Jack mentioned, there are some obstacles in terms of familiarity with the standard operating procedures, data quality practices. But this is something that other states have implemented. They've provided the opportunity to get trained on the SOPs and, potentially collect data in a way that's contributing to our water quality monitoring and assessment programs. Right now, we just don't have the staff to implement those type of programs, but it's something that we're trying to find funding for going forward. So we've kind of developed the broad outlines of that type of program. It just hasn't been possible to really implement it on a widespread basis. But if that's something folks are interested in, I'd be willing to talk to them about it. We did a survey of external organizations just before COVID hit. I think we sent a survey out to over 1,000 organizations that trying to gauge their level of interest in participating in an external biological monitoring program, and we received a very positive response. So we were able to take that to our leadership and demonstrate that the interest is there if we can just find the funding to put this together.
Cool, that sounds great. Yeah, I love that idea of, having that training and, maybe certification piece of it, so you feel a little bit more empowered to let the community help. So cool.
So I guess I should speak up here that, so the Izaak Walton League has a Clean Water Challenge that used to be called our Save the Streams program. And we developed the macroinvertebrate testing and that kind of thing. And EPA embraced it, so they've used our protocols for decades now. But they now have a test for salt. So there's little strips of paper that you can use and monitor. We're working with PCA on that program. And there's also been some chemical testing. So I'm not sure exactly which things are being tested. I know down in Austin, they were doing chemical testing for E. coli and some of those kinds of things. But we have like what we call a clean water hub. And so if you have somebody take a picture, a selfie, so you got their coordinates, and then with the results from whatever testing they have done gets sent into this hub, and then they can spit back information, so you can track what's going on. I know I went out and looked, and somebody turned in some information for Starring Lake in my watershed district, and it was a chemical. They were doing chemical testing there that turned up on that Clean Water Hub. So there might be a possibility. I mean, maybe we could help come up with some money. We could have somebody come out from national office and do training. And so it's something we should maybe talk about. But Jack's right. It's I know we've done a lot of restoration just on. My Izaak Walton chapters in on Bush Lake, and we do monitoring. We've done monitoring of that for years and turned that into PCA. What was I going to say? Anyway, but I just wanted to put in a plug for Jack and a thank you, because he's been, with his ecology background, that's so different than what you normally get. And so he's been able to come out and help us take a look at where we've got cattails and what's native and what's non-native and what should we be looking at. So I think there's a lot of partnering that can go on, but, I know what I was going to say. So working with volunteers it's not easy. You've got to be really organized. You got to have specifics as, okay, you're going to do this. So it's like we do that with our restorations. Okay, we drill this hole here, put this plant in there. It's almost like you got an auto line, manufacturing line going, but there's got to be really good supervision. And and somebody's got to be really organizing it. So, but I wanted to shout out a thank you to Jack, because that ecological background's just brought a whole new dimension, I think, to Bloomington and helping us look for a path forward. So this has been really helpful for me to understand what my own city's doing. So.
Are there any other questions, comments, you know, things you want to work through in your own community? You want to use the rest of our time up?
I guess I want to ask Jack, so have you shared your ecological framework here with my watershed district?
With Raleigh Perg?
Yeah.
No, because that one was for Penn Lake, which is in 9 Mile. So 9 Mile knows pretty much all about it. We're partnered pretty heavily on that particular plan. I'll make them for any specific project we're working on, but we're doing a big Penn Lake rehabilitation initiative. So I haven't, but I mean, I didn't invent that method or anything. So it's not like I don't know. They're pretty common out there.
So are you part of our technical advisory committee, or is that somebody else in the department, your department?
It probably, I haven't heard from you all in a while, I'll be honest. So
Okay.
I don't know.
Well, maybe it's Brian. I don't know.
Yeah.
Used to be Steve. We do a lot of work with 9 Mile. So that's why I was curious as to, we're trying to figure out whether we could extend our boundary just by doing something jointly with them too. So, but we're starting up more educational kind of stuff. So it'd be good to find out how we can take what you're doing and make it something available for the city too. So
yeah.
Anyway, but thanks for all you're doing.
Well, if we don't have anything else, I'll share a link in here for our next upcoming workshops. We have a couple of smart salting workshops coming up here. Available. Oh, here, I'll give you the other link because this one doesn't have all of the links. So we talked about some salt and chloride impacts to water today. So that is potentially of interest. One is specifically for local leaders. So it is about training decision makers in communities, mayors, elected officials, commissioners on smart salting considerations in your community. So I definitely encourage that. After that, in December, we're talking about electric vehicles and how to make your communities smart and EV ready. So more to come. Thank you, Jill, for coordinating. Thank you, John, Jacquelyn, and Jack for speaking. Yeah, this is fun. I hope my next workshop has everyone's name starting with the same letter as well. So really appreciate your time and the work that you're doing to protect our waters across Minnesota and in our communities. So anything else, Jill, to wrap up?
Nope, I think that's it, but thanks for helping pull this together.
Yeah. Thank you, everyone. We'll send out slides in the recording in the next week or so.
All righty. Take care,
Thanks.
everybody. Thank you so much.
Thank you.
Thanks.
It's nice to meet you.
Thank you.